All right, everybody, let's go ahead and pick up here with part 2 of our discussion of infectious disease. So last time we looked at really the main topic is the chain of infection and how breaking that chain is essential for disease control. Now I want to talk about next is the science of epidemiology. Epidemiology, everyone, is not studying a disease per se, but basically where diseases occur and how they're spread. And epidemiologists look at transmission not just from one person to another, but through an entire population. A little history: John Snow, pictured here, is considered the father of epidemiology because back in 1855, he was a British biologist, by the way. He publishes the first epidemiological study where he proves that cholera is spread by contaminated water in one of 2 wells that were supplying London at the time. After treating that water with chlorine, cases dropped by over 50%. So he is considered the father of epidemiology. Today, the Centers for Disease Control and Prevention, or CDC, are responsible for really epidemiology, okay? They track data like this, looking at Lyme disease by month, and are just responsible for that in general, okay? Now, the CDC categorizes diseases different ways. So we have communicable disease. Communicable disease are those that can be transmitted from host to host. So it can be transmitted from another infected individual, directly or indirectly. Contagious is something that's highly communicable, a disease that's easily transmitted. It's highly communicable from host to host. Non-communicable diseases are those that arise inside you or from normal microbiota, opportunistic pathogens inside you. They're not spread from one host to another. Non-communicable also includes diseases not caused by infectious agents. Cancer, diabetes, genetic disorders, etc. At least most cancers. So it could be a disease that arises inside the host or from the host's normal microbiota, or something that's not caused by a microbe. Then we look at where the disease occurs. Endemic means the disease is normally present in a region. So it's normally there. An epidemic is a disease that's present in far greater frequency than usual. So it's an endemic disease, but it's in greater frequency than it should be. That's an epidemic. A pandemic is an epidemic on more than one continent. And then a sporadic disease is one that occurs very irregularly or rarely in a region. So we can look at it like this. So endemic, so the beige color here, the pinkish beige color here is the normal range. And the red dot, that's a new case. So notice it's endemic, this is normally there. There's a few new cases, but normally there. Sporadic, oh, here's a rare disease cropped up here or here. Epidemic, look, it's normally present, but in greater frequency right here. So there's an epidemic of something there. And then a pandemic would be this greater frequency on more than one continent. All right? Makes sense? Now, we also have, and this can be important from a medical perspective, nationally notifiable infectious diseases. These are those that must be reported to the CDC. If a case comes up, hospitals, physicians, or healthcare workers must report to the CDC. As of 2016, there were 70 such diseases. The list has grown a little bit, like COVID's on that list now. But here we see what some these are the nationally notifiable infectious diseases that you are required to report to state health departments and then eventually the CDC. All right. Another interesting thing that the CDC puts out to, and you can actually subscribe to this and get this through email every week, is the MMWR reports, Morbidity and Mortality Weekly Reports. Now, these are weekly CDC reports by state on the different occurrences of disease. Okay, particularly these nationally notifiable ones. Now, just so we're on the same page, morbidity refers to the frequency of diseased individuals. So how many people have the disease? And mortality is how many die from it. So the report might look like this. So like it says United States reporting area, New England. So here's some states. And these are your diseases, giardias, gonorrhea, okay? And then it tells you the number of cases. All right? This is kind of neat if you're interested in that. Now, what about effects of climate change on infectious disease? So this plays right into epidemiology. All right, basic concept, everyone. Due to climate change, the number of incidences, cases, and the range of disease-causing microbes, pathogens, are expanding. Both the number of incidences, the number of cases, and the geographic range. And this is primarily due to rising global temperatures worldwide. Pathogens are also being released from thawing permafrost. We talked about that in a previous chapter. Many of these that are being released are unknown. Remember, they've been frozen in permafrost for thousands of years. Here's a recent article. Anthrax outbreak in Russia thought to be a result of thawing permafrost. Warming bodies of water play into this because they promote the growth of pathogenic aquatic microbes. And it expands the range of where these pathogenic aquatic microbes are found. A good example is Vibrio species. Now, Vibrio cholera causes cholera. They're already growing faster in response to rising marine temperatures, can cause significant medical issues, wound infections, cholera, foodborne illness, even sepsis. Okay? And then going along with, we're still under warming bodies of water here, everyone. Going along with this is toxic cyanobacterial or toxic dinoflagellate blooms. Remember, cyanobacteria is a prokaryote, sometimes called blue-green algae, and dinoflagellates are little single-celled eukaryotes. Both of these are types of algae. All right? Now, what we mean by a bloom is a huge increase in population numbers of either cyanobacteria in freshwater or dinoflagellates in the ocean, in marine systems. When you get this huge explosion in the population of these organisms, they use up all the oxygen in the water that they're in. So it creates anoxic or very oxygen-poor conditions for life that lives below them. And that seriously disrupts ecosystems, can lead to huge fish die-offs, for example. Now, both cyanobacteria and dinoflagellates grow better in low nutrients and higher temperatures. They grow better in those conditions than other phytoplankton. And the toxins they release, besides using up all the oxygen, they release toxins, can cause liver, digestive, neurological, or even skin diseases. Example of a big algal bloom, cyanobacteria, Lake Erie back in 2011. All this green is the cyanobacterial blooms. Okay? Very, very problematic. And then also, as you might imagine, with rising global temperatures, we see an increase in vector-borne diseases, both in their distribution and their range. Now remember, vector-borne means carried by mainly insects. Now, many vector-borne diseases are migrating north to areas that were previously too cold to sustain those insects. Insects are heavily dependent upon environmental temperature. That's what ambient means, the outside environmental temperature, water availability, and humidity. So as it warms up, insects stay alive longer and do better at those warmer temperatures. And their incubation times are shorter. In other words, they can reproduce faster in warmer temperatures. So the spread of these diseases is being monitored by the CDC and the World Health Organization. And we see examples here: malaria, dengue fever, encephalitis, West Nile virus, even plague. Their distribution's expanding and their range is expanding, as are tick-borne diseases like encephalitis again, Lyme disease, rickettsiosis, and schistosomiasis. Another one is chikungunya, is another one that's expanding. Now, this has typically been a tropical viral disease, but it's now being reported in parts of the United States and even Europe. Okay? It's spread by this mosquito. All right, so it's vector-borne. Causes fever, joint pain, headaches, muscle pain, joint swelling, and rash. Good news is it's typically not that serious. Most infected get better in a week or so, but in some individuals, they can have really, really disabling pain for months. All right? Prior to 2006, it was rarely found in travelers, but now becoming much more common as that disease migrates to previous areas that were previously too cold. You see there's even incidences here in the United States, the dark blue, locally acquired, and travel-associated cases. Look at that. All right, even in Alaska. Okay? Now, the last major topic I want to talk about here in this chapter, and it's certainly relevant to any of you going into the healthcare industry and nursing, is healthcare-associated infections. And everyone, you have to know this terminology. Healthcare-associated infections are called nosocomial infections. So these would be infections picked up in a medical or healthcare facility or from a healthcare worker. Nosocomial infections. Factors influencing nosocomial infections include the presence of microbes in a hospital or other facility, how many immunocompromised patients are there, and the relative transmission of pathogens between staff and patient, between staff, and patient to patient. So if you look here, what kinds of microorganisms are in the hospital environment? What about immunocompromised patients? And what about transmission? And where those all overlap, this is where you get your healthcare-associated infections or nosocomial infections. Types include the following. I'll just show you this visually. Look at this pie chart. All right, as you can see, everyone, most nosocomial infections are UTIs, urinary tract infections. But then we have others, including skin infections, surgical site infections, respiratory tract infections, and bacteremia. That's bacteria in the blood. Okay? So how do we control nosocomial infections? Now, everyone, you're going to control nosocomial infections the same way you're going to control any other outbreak. You have to break the chain. All right? How do we break the chain of transmission? Let's look at each link in the chain. What can you do about the causative agent, the microbe itself? Rapid identification of the microorganism involved so we know what we're dealing with. Prompt and proper treatment then. Decontamination, and we've talked about that in previous chapters. What about the reservoir? Okay, to control things at the reservoir level, good health and hygiene, sanitizing equipment and surfaces, and then disinfection and sterilization where necessary. How do we slow down the mode of transmission? That's a big one, okay? frequent hand washing, hand hygiene, airflow control. Again, if needed, disinfection and sterilization, proper food handling, isolation precautions if necessary. How do we deal with the portal of entry to break that link? Aseptic technique, all right? Wound care, and especially that aseptic technique to avoid things like parenteral, that parenteral route. Hand hygiene again, and catheter care. Remember, UTIs are the most common. How do we break the chain for susceptibility of the host? Treatment of their primary disease. And recognizing high-risk patients, those that are immunocompromised. And how do we deal with the portal of exit? Proper attire. Again, hand hygiene. Note that theme here. Proper trash and waste disposal, and proper control of secretions and excretions. So you can see that here, these boxes show how can we break the different links in this chain. And remember, to control a disease, an outbreak, whether it's nosocomial or not, you have to break a chain, a link in this chain, or more, one or more links in the chain. Okay? All right, everyone, that's it for the part 2 of the chapter on infectious disease. The next chapter, and in fact the rest of the course, we're going to look at examples of various infectious diseases and their clinical and medical importance with an eye on this chain of infection. All right, everyone, I hope you have a nice rest of the day, and until next time, bye-bye.